Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Hello, today on the Microscopists, I'm joined by Timo Zimmermann, a very special friend and a team leader at the EMBL. And we talk about how an EMBO microscopy course changed my entire career path thanks to Timo.
An EMBL course some years before actually changed my career path completely. I would say changed my life in this aspect, and that's why for me this was like coming full circle.
His recent 3D reconstructions from data collected in 1996.
Volume renderings, I think I read it recently, so this is actually done with Fiji. Basically the original data sets and also 3D renderings with shadings I did then. I just redid them once because the shading and the color scheme is now a little bit better.
And how Star Trek helped him learn English.
I did my school English paper that was part of my final examination on Star Trek, and I got full grades.
All in this episode of the Microscopists. Hi, welcome to this episode of the Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Timo Zimmermann from EMBL. And before I go to Timo, I'm going to embarrass him just a little bit. Now he's panicking. Yeah, one of my questions for the guests, so Timo, pre-prep here, I'll ask her who are their inspirations, or who's been their inspiration. And if I was ever a guest myself and I was asked who's been my inspiration in my career, one of the first names, if not the first name that would pop into my head, is Timo. So it's a great honor, Timo, to have you here today as a guest. So Timo, how are you today?
Great, and thanks for having me. And actually, yeah, thanks for the compliment. But, I don't think it's deserved.
No, it really is. I remember meeting you back in 2001. And at that meeting, you sent me a picture of this meeting. So I'll bring this up while we're talking about it. I can't believe, and this is the first, I only saw this picture today. It was like, oh my goodness, this is the crowd. It was an EMBO course. I'm not even sure everyone's on there because I'm sure there was more of us. It felt like a big crowd. It was 10 days, 6 days a week course. You were the lead demonstrator on the Leica, SP2 at the time.
Yes.
With Spectral, with AOBS, starting to do FRAP and everything else. And it was the way you taught and showed that there's a career in helping others do microscopy. And the way you trusted us, naively, to use it might it was, you showed there was a different path, not just postdoctoral research. It's academic, but not academic, if that makes sense.
Yeah, I mean, I remember the course very well. And actually, first of all, I remember our discussions during that course very well because you were just about, or you already had accepted, I think, the position and were just embarking on this. And therefore, I saw a lot of similar challenges for both of us, and so I think this was really very interesting, and I really enjoyed my time at this point. I mean, I think I was exactly in the kind of place I wanted to be, career-wise, and actually, I think even to this day, every now and then, because for example, these days I show a lot of people around in the imaging center where I'm working now, and I realize I'm actually still really enthusiastic about certain things. And I'm very much in line with that. And I think if that came across, then that's it. Yeah, I felt like that then.
It was your enthusiasm that was contained. I don't know if you remember. The sessions were quite long, and you'd have to go for a toilet break every now and then, and you'd leave us to carry on, use the instruments ourselves, which was very brave. And I'm not sure you ever came back and we hadn't crashed the system whilst you were gone. We were constantly crashing it in your absence. I don't know if you recall that.
Not even as a bad memory.
But I remember your response going, "Oh, guys, how did you do it? So you were taking it almost like a learning objective of what we were doing to sort of kind of mess it up in a way that hadn't been done before. And that was Oliver Rox was probably my partner in crime in a lot of this, and Claudia Lucas probably in hysterics as we're trying to push it to its limits and finding the limits at the time. So that was super cool. So was that the first course you taught you had been a tutor on?
Exactly, and that I think made it very important for me because an EMBL course some years before actually changed my career path completely. I would say changed my life in this aspect, and that's why for me this was like coming full circle to be instructor in this course, and especially in the first one, because it was really I kind of arrived. Yeah, I mean that's also why I sent the picture. I mean, this is an EMBL course in Lisbon from 1994, so 7 years before, and I'm one of the circles in the back, but there's a lot of other people that we know together. And I mean, at a time when one hardly remembers, but this course exposed me to confocal microscopy at a time when it was a really, really rare modality. And it moved me from electron microscopy, where I was really enjoying myself, to confocal microscopy. And by this really changed my career path. And so for me, this was fundamental. And this was one EMBL course. So this one course can really, my apologies for the very fancy illumination system that now gives nice light effects. No, it was really, this changed a lot for me. And therefore, I was super enthusiastic to actually go and be an instructor in these courses.
And so I think that's quite something. For me, the course, I would argue that the course did change the direction of my career, so therefore it has changed my life. Because I don't think I would have gone into this position necessarily. I don't think I'd have done what I've done within that position if I hadn't seen what was possible. And so the EMBO course and EMBO courses, both of us, have been really inspirational. And I noticed one of these circles over my shoulder just here is Rainer Peppercock. Is that right, Rainer, on that?
I think I mean, he was at this course, and I think I circled him correctly. It's a very small picture, but I think that was Rainer. And first time I met him, and of course, Rainer is an extremely important person in my career. Was and still is.
Yeah, because not only at that point, but in that first picture that we had, that 2001 course, of course, Rainer again is on here, just sort of.
Yeah.
So he actually set up the course with Philip Bastians. So he, these courses and the legacy of people running them and taking them forward, it does change people's trajectories. I don't think we should underplay the importance of these EMBO courses and other courses and events around it, and just how big an influence that they can have with that.
That's basically it. And I mean, I can, if you feel like I felt about this initial course, I basically understand it, because that's what can happen.
So I'm going to take you back. You said you did EM, but let me go back, way back. When you were, I know, a small child, what is the first job that you could remember ever wanting to do?
Astronaut.
An astronaut.
I really wanted to be an astronaut, and I think I gave up on that only very late, even after studying and even after the PhD. At some point, I didn't follow it up anymore. I even learned scuba diving because I thought it would be a good, let's say, skill to have. So I never ruled it out, because astronauts have a scientific training, so it was not impossible. Yeah.
You still want to go up into space?
No, I think that has passed. But let's say an active research career, partially also initially I very much like fieldwork as well. But it's not the only thing. I mean, I think I really enjoy the things I've done and keep doing now. So there's no regrets. But if you wonder what it was, I wanted to be an astronaut. It was a very, very clear wish.
So actually, my next follow-up question is usually, when you started your degree, what did you want to be? And so I think you've answered it. It's still an astronaut at that point. So what degree did you do? What was your
Sorry,
degree? What was your degree?
Biology. But maybe again, also saying about that one, or how one can follow one's intuition or one's likings. I was involved in imaging right away. I started biology, and I really, I think it influenced me in many ways. But from the first semester on, I worked in the electron microscopy laboratory and became a part of it. Basically, at a time when nobody should be in this kind of lab, but I managed somehow with a very supportive professor to work my way in, and I never left. And so I think the way that I approached biology was very much influenced by this all through. But in addition, of course, I very much enjoyed some of the projects I did then. I think by now I mean, I still can consider myself a trained biologist, but I don't do biology anymore. I think I have to be realistic about that, because it's a field that requires so much dedication and learning continuously, that this is not my field anymore. But
No, that's very true, actually. And biology, yeah, from our degree days, has moved on significantly to keep up. But so here's a question. So you're a microscopist. I think it's a good description of your skills. You're a microscopist for life sciences predominantly. How can you be helping users with a microscope if you don't understand their cutting-edge biological questions?
no, I think this should still be possible. Although even that's, I mean, the fields have really moved on a lot, and are being transformed time and time again. I think we witnessed some of these transformations: green fluorescent protein, polymerase chain reaction. Even, I mean, I would say fundamentally changed the world. CRISPR now, but I think we do understand enough to actually understand the challenge, at least the outline of the challenge. And what then is I think part of my job, part of your job, is to be the interpreter between the biologist and the research question and the instrumentation. That's how I always saw myself, like to be there to connect these two parts.
I no, totally agree. It's always got to be careful who's listening out of my user base, but you'll get people coming, they'll tell you that their whole protein pathway, and at the end of it, my question is, so what colors have you got, and where are we expecting to see them? Because that's what we can enable you to do. And then we tell them, so what are you looking for? Protein interactions, you're looking at dynamics, you're looking at protein. So then you can start putting your dynamic studies, your FRAPs, your FRETs, your FCSs, and everything else into play. But
it's true, yeah. I mean, this, you have to find, let's say, the common level on which to exchange, and it's very often the case. I mean, I think we have all had these project discussions where people basically give you a lecture on their field, and it doesn't involve imaging at any point, and then after 15-20 minutes you finally cut to the chase. But still, I mean, important is to find that common ground and then to work on that.
So from your degree, you went into a PhD. Where was your PhD done?
So both my initial studies as well as my PhD were done in Munich, which was a bit of a surprise, because I think, as I said, since in 1994, more or less towards the end of my studies, I was exposed to confocal microscopy, and therefore one of the few people that had some practical knowledge. I got the possibility to work with a confocal microscope. I actually had, my initial plan was to go to Canada to do functional neuromorphology. That would have been very imaging based. That was actually my initial plan. And so I stayed in Munich for the installation of the first confocal microscope in the institute, which I then also used on some of the first variants of fluorescent proteins in a developmental biology setting, so using cellular slime mold, both as a single cell organism, so looking really at the single cells, and also at the multicellular aggregation stages.
And you sent another picture with actually another guest that we've had. So this is, and it says PhD crew. Was this during your PhD, or was this after your PhD?
These are the people I did my PhD with.
So, okay, Kota?
Yes.
Jens?
Yeah, Jens.
It is Jens.
We are all from the same group, Munich.
Wow. And I don't know the lady next to you.
Yeah, she's not in imaging, at least not in microscopy. I think in the end she did a lot of remote sensing. So because our PhD supervisor, he was working on cellular slime mold, but one of his contributions to the field was together. So his name is Florian Siegert, and together with Kees Weijer, they were some of the first that actually did image processing on these developmental mechanisms in dictyostelium. And that's what attracted all of us, actually, the imaging aspect. And Florian, in his career later on, changed to remote sensing. Actually, more or less already at that time, he had a very strong interest in remote sensing in Indonesia for deforestation. And he applied the same kinds of methods. And of course, with twists, I mean, you needed these GIS referencing systems and whatever. But he applied image processing to ecology. And that's where he, in the end, also got his professorship. So meaning he changed fields because the tool is extremely variable as long as you are applying it in a good way.
So I'm just amazed that that's 3 in one group that are still in the imaging community and still very influential and big names, all of them in their own rights. And I quite like this picture as well. So I've got to ask, what was Kota like as a roommate?
Well, he was not my. Oh, actually, we did share an apartment together later on. No, the thing is, Kota just came from Japan when we started. So he had just arrived. I just came into the group. So we basically met on day one, and I mean it was a really fun time. Always, I mean we overlapped at the PhD. We had a long time together as well then in Heidelberg, EMBL. So meaning this really helped. Same with Jens. I mean, this was basically Jens, me, and Kota. We all went to the ALMF, and this really influenced us for a long time. And that also showed maybe what kind of profile was at this point needed for these things. And this is what we had. I'd like to say 2 things in this context. First of all, Florian, as our supervisor, I mean, he had set up a lot of imaging rigs, and they were connected with a lot of cables. And we spent, I would say, half of our PhD under those tables. And he was saying, this is the kind of skill that will really help you in your careers. That basically this hands-on thing that really fixing the systems. And he was very right. And the other thing is, I mean, many people in the field that may also see this discussion. They know Kota in different forms, and they realize he's an outstanding image analyst and a very inspired one and a very interesting person. I think what very few people know or can remember is that Kota is the best experimentalist I ever met, but he stopped doing experiments. And again, a person like Florian changed the field. But I mean, he did outstanding dictyostelium experiments. Even before that, in Japan, he made a single amoeba crawl through tunnels. So I mean, he has this extremely systematic, well-designed experimental approach that I actually don't have. So that was funny. That Kota is actually an outstanding experimentalist.
I didn't know that, and I take it this is. I might as well stay with your PhD just briefly. This is your original workstation during your PhD?
Actually, even before, I used this for EM reconstruction mainly. It's a RISC workstation that was running under Unix, and at this point, the tools were still very limited. I used Data Explorer and actually also, I think even on that one, interactive data language, which I then used for everything afterwards. I did most of my rendering and analysis also for the PhD in interactive data language.
So how much? I have only just realized. So you sense, when you look at EM, image restoration, reconstruction, rendering, most importantly, you get some stunning images. Now, if you're watching on YouTube, you'll see some of the rendered images. If you're a new to electron microscopy, and you're looking at the image, and we're thinking, that's a bit clunky. But oh my goodness, I didn't realize that this was 1996 that you rendered images, which is unbelievable.
Yeah, I mean, I must say there's maybe one disclaimer here. The volume renderings, I think I read it recently. So this is actually done with Fiji. Basically the original data sets and also 3D renderings with shadings I did then. I just redid them once because the shading and the color scheme is now a bit better. But the work is based on I didn't do any more reconstructions. I just used the reconstructions I had and re-rendered them once. So
Okay. Yeah, wait a minute. I'm catching up now. You've re-rendered data from 1996. What was the data stored on that enabled you to still be able to access it? Because in 96, you were talking floppy disks.
CDs. CDs are still good.
Only just on CDs.
Yeah. No, I mean, I basically backed up on CD. And the reason why I revisited the data is that I think maybe biologically, this was the thing that inspired me most. This was functional neuromorphology, and it is about how to connect or how individual system of insects, the neural cells that are underlying the facets are actually connecting. And this was an evolutionary riddle that I really wanted to crack together with my colleagues. And it's such an interesting story that was only half told. So I never could really show all the morphological data that we had, all the synaptology. So many years later, I realized whenever I was talking to people that are having an evolutionary interest, that this is an interesting evolutionary story. So every now and then, every few years, since 25, 30 years, I'm retelling that story. And that's also why I re-rendered the data at some point, because they looked dated, even though what they were, this was very much aligned with research interests in genelia at the time. So meaning this was interesting data that I wanted to share in a bit more fashionable way. So every 5-6 years, I give a talk about this just because it's an interesting biological question.
I'm amazed the CD still worked as well. That's quite something. So, what countries have you? How many different countries? It's just Germany and Spain that you've worked in. Because from your PhD, you then went to the LMF. Is that right? To light microscopy facility.
Now, after the PhD, I went to the advanced light microscopy facility in Heidelberg, the EMBL.
And then after that, because EMBL, you can only work so many years. Is that correct?
Mm-hmm. Yeah. So you'd have in principle a time limit, and I was aware of that. And I mean, I was having a specialist position in the ALMF. I had no intention to remain, because simply for that reason that it ideally helps you to qualify. And I think I really enjoyed my time there. I mean, I was in the end there for 6 years instead of the maximum that would have been possible would have been 9. But I know that my motivation was basically to go on my own terms, not at the last moment. I mean, I really would have enjoyed another 3 years. I mean, the projects that I was involved in I really liked them, like working on malaria movement, working on initially also on motors of vaccinia virus. There was always something. Then the method development. I mean, I was working a lot on spectral imaging at that time. The exposure to all the new technologies. I mean, this was ideal, but also at some point, of course, it qualifies you for maybe further steps. And so when an opportunity came along to start a unit in Barcelona, I went for that under this feeling, okay, I should not. I mean, you have to go on at some point, go where you want to go. And I think I did.
And your CRG team grew and grew and grew once you went there. And you, again, for those, I would encourage people to go and have a look at some pictures off the YouTube at the very least. You started with just the two of you. And then that obviously grew to quite a sizable team.
Yeah, it grew and shrank a little bit because I think what's also very important is sustainability. And I did not manage to fully establish image processing as a backup. Others, I think, succeeded more. But this was really also, I didn't want, I mean, I had really good colleagues all the time. I mean, these are very, very dear to my heart, and they know it. And I mean, one sees it over the years. I mean, it's, but it was also very important to look out for, of course, for career planning. And I just realized that, like in the middle, we had one position more, which was time limited, and there was, it was not. I didn't want to risk to promise, okay, this will be self-sustaining, and then it isn't, because that's of course you're messing with other people's career. But I think I had we had a well-staffed facility that was very much grown to the size of the needs that we had. I mean, maybe whatever, 13, 14, 15 systems, 250 users. So this kind of thing, I think this was very well matched, and still is, I hope.
And the team themselves, I know some of the team, they get out there at the conferences. So you obviously were helping develop their own careers within the core facility as well, which is super cool. I asked you earlier about what your dream job was when you were young, and it was an astronaut. Today, I'm going to guess if you could choose any job in the world today, what would you choose?
Okay, since astronaut is off the table, actually, I mean, this may sound weird. Actually, no, I'm really okay.
That is the right answer.
If you could just maybe take this imaging center and transplant it to the beach, I'm good. But that's it. And that wouldn't really work for technical reasons. So therefore, I'm really good.
No, that's a good answer. So here's a slightly different question. If you could sample any job, any different type of job, and I'm not going to let you choose an astronaut for today, any other type of job, what type of job have you ever thought, gosh, I'd love to know what it's like to be in that position, just to feel it and see, for whatever reason, what sort of job would you like to just sample? Not to do as a career, but just to sample.
I don't think I'm qualified for that. So basically, I couldn't do it. But I mean, maybe one dream I would have is at some point some kind of time out to really think deep about the problem. Maybe the way that theoretical physicists are sometimes doing or have done. I think that's maybe, and I have to, of course, the disclaimer is I am not a theoretical physicist. I don't have the tool set to do this, but I'm just reading a book by Heisenberg, for example, and I mean, I just think this focus on a singular problem, I would like that, but it would need to be in an environment where you can really focus on this. I had moments like that. I mean, I have a Finnish side of my family, so I have connections to Finland. I spend these days fairly little and maybe more limited time on the This is the view from the estate of my family, like with their little houses, and this is the view. And of course, this can allow you to really think deep about the problem. And I did it maybe twice in my life. I did it once for this neural connection problem. I remember I was really sitting there and I had the time, especially in Finnish summer, where the days are so infinitely long that you have the feeling you have all the time in the world. I was really making progress then. I understood the problem and the solution. And similarly with illumination regimes for single and multi-beam scanning systems. I mean, these are fairly basic equations, but I could calculate the whole thing through in such a surrounding. So basically, this kind of reclusion and working on a theoretical problem, I think that's really that would be what I would like to sample.
Okay, so you wouldn't want to be a rock star, or a classic guitarist, or anything else?
Not really. This is.
Do you still?
This is only to attract girls, actually.
No. So I'll put a picture up, which is throwing Timo a bit of him playing a guitar on a marine biology course.
Yeah. Which was very important for me. No, I mean, first of all, I play very bad, and I can only memorize tablature like small pieces, maybe 4 minutes tops, and that used to be a long time ago. But it was a very nice surrounding, and it was actually really inviting to maybe also take a guitar along and enjoy the scenery. This was in Croatia. I did these marine biology courses with the group where I was initially working for many years, and this was always a very special moment or experience, because it's exactly this connection to the field, and maybe something also to appreciate when you're snorkeling, for example, the density of organisms, which if you are looking at how the questions are posed in a lab, is always reduced to a single mechanism, to a single specific question. But seeing these things in context was always very, very important. So whenever I think about a biological issue or even about probes, tools, I mean, we're basically using fluorescent proteins coming out of cnidarians. very few people that use them as a tool, very few people that develop them as a tool actually take the ecology into account. And I mean, the guy that found the living fossil, Fricke, Hans Fricke, I mean, he went around with a self-built submarine, and he found like this living fossil, this coelacanth, that's like a transition to the land animals in the depths in front of India. But he also, for example, in the 90s or in the 80s, was already like as a side comment commenting on deep sea reefs, and that they have deep sea corals, deep water corals that have blue fluorescence to probably support the endosymbionts. First of all, ecologists always knew that it's not the algae that are colorful, but that it's actually the animals. And all of these things that let's say lab biologists very often even quote wrongly when they talk about corals or whatever, it's all known. And if these groups could talk more, they would understand better also how to optimize some of these, of where to find the next class of compounds. Communication is key, but it's always missing between the fields.
So 94 was quite something, if you had the EMBL course and the marine course as well. So that was quite a year for you. When did you first meet your wife?
At EMBL, actually, at many years later. And she's from Croatia, but I know the area where she's from very well from the marine biology courses, but we met in Germany.
Okay. And I'm going to ask, how many, 2 children, yes?
2 children, yeah.
And either of them following in your footsteps scientifically?
I don't think so. I mean, it's way too early. And that's maybe the other thing. We already touched on it twice, how you can completely transform your career. And I wouldn't expect them to know I mean, one is 14, the other one is 12. I mean, even the older one, I would say, has at least 10 years time before they have to get really serious about, I mean, they should not idle away their time. That's not the point, but there are choices.
But they still want to be footballers.
Honestly, these days, I wouldn't know. It keeps changing. We are hoping one of them will be a basketball player because that will bring in money, but you know, these things never work out.
Talking of sports, a couple of things. Firstly, I quite often when we meet at different conferences, such as ELMI, we will go for a run in the morning, and I've got multiple selfies of us running. And my panic for today, I could only find one, which is really poor. It's not even the best one. I can put myself with my hat on. There you go, on it. There you go, I can just wear my hat. So this is us in San Francisco.
San Francisco, yeah.
In San Francisco. But I should have pictures from Portugal. We went out on I think that was the first time we possibly went running to. Oh no, first time we went running. I remember where that was. Oh no, can you remember the first time we went running together?
I mean, I would have also said Portugal because I have a specific memory about Portugal. But let's see, if you think about, must have been ELMI meetings. The one before would have been in Sweden, and then the one before in Spain. I think it was Portugal.
I think it was Semmering, Austria.
Yeah, but this was, of course, it was. That's the one. Oh yeah, up and downhill.
Because I don't know. There was a bigger group of us that went running, and we
went with several people, and that was the ultimate hangover cure. For some, for others, it was basically maybe just pointing out that they should stay in bed. Oh yeah, that was a good one. But it was a really good running area. I mean, with the elevation and the downhill.
I can't remember one of the. I remember us running. I can't remember all the other runners with us, but there was 2 female runners, and one of them, my goodness, she was a monster. We were going round the hill, and she said, "I'll meet you," and she just turned vertically up it and just ran up it. It's like,
puts us in our place.
Yeah, really, she was just quite an incredible runner. I think she was actually properly nationally or internationally competitive as well. That was, I remember that, and of course, Portugal for ELMI was on the beach.
Nice on the beach, yeah. I mean, for me, I have this memory that since Portugal is already in the UK time zone, I actually got up 1 hour too early. I mean, anyway, like we're setting out early, like around 6 was the plan. So I was sitting in the lobby at 5 because I didn't realize that I was in the wrong time zone.
Yeah, I also remember ringing you up when you missed your alarm once.
Yeah, that could be. There was probably a reason for that.
Which would be the drink the night before again. Back to the same theme. But it's not just that. We also your football. I mean, think about conferences. And I'll ask a question about this in a moment. But you are part of an annual stalwart of the academic team for the ELMI academic football team itself. With Timo position. Go on, Timo.
Yeah, no, I mean, I think this is actually one of the nicest activities that now also is established. Especially this photo, I think shows it even better. So this is since 2015. I think every year the ELMI conference, the European Light Microscopy Initiative conference, has on the first day a game between the academics and the companies, and well, normally we get slaughtered. Let's be honest about that. But I think it's such a great thing because, I mean, if people can see or recognize some of the faces in the back, these are different companies. We're of course from different institutes, and we're all in the game together. And that's, I think, symbolically, that's exactly what's happening. I mean, we share this deep interest in this technology field, and we have so much that connects us. And this is actually shown in this moment of good spirit. So for me, these football games, they're really fun. They're not fun to watch, I think, because we are not really good, I guess. But they are really they're a nice gesture.
The next year it's ours. But a shout out actually to Florian, who I think really got this off the ground, and Bulont, who's carried on the torch for Olympus stroke Evident. And it's great seeing the likes of Zeiss and Leica and other Nikon actually turning up and playing as colleagues.
Yes.
Which is, you think this great rivalry between these companies, and actually, they're just like academics. I guess academically we compete against each other, but we also collaborate. It's maybe less on the company side, but in this point, yeah, they'll all play together. They'll have a drink of water together and an ice cream, and then.
No, and that's really what makes it nice. I mean, this idea that came up like for this ELMI meeting in Spain at the time in Sitges. Yeah, it came basically. I mean, it was Florian, very much so. Florian, I mean, at that point at Olympus, now at Leica, and many, yeah, and it just caught on. It was such a good idea.
And just one other exercise. So during lockdown, what exercises did you pick? Because obviously you were in Spain, so very limited on what you could do outside. How did you try and keep fit at home? Where
I basically, I mean, I tried to work out in the house and in the apartment, and because lockdown in Spain was harsh. I mean, if I just think at the level of the children, but I tried to run in the apartment. I tried to actually then do chin-ups, and this is when I fell out of the door frame and completely smashed my elbow. Actually, I should have added but then viewer discretion would be advised. I should have added the X-ray of the fracture, which was really massive. Like the elbow just came off, and I didn't realize for another 2 months.
So you were doing a chin up.
Yeah.
I guess you had some contraption on the door frame.
Yeah, which was unfortunately permanently fixed. So when actually I went up to energetically, and I just unhinged it, and it came down with me under full tension. So actually, does one still see the scar? Yep.
Got a big
there
scar
it is.
on there.
Basically, it makes me look like Wolverine, and I mean inside it's just titanium.
Thanks for you're not that much like Wolverine. Thanks for shaving. You're not that much like Wolverine.
Yeah. No, but it was a massive injury by trying to stay fit. So basically, I achieved the opposite. It took me, I think, a long time afterwards. And now I'm more or less back in good shape. But
so you came down, you hurt yourself. It took 2 months before you went and got it X-rayed and realised that your elbow was completely in pieces.
Well, I think we realized before that there was a fracture, but they thought it heals on its own because I mean, for some reason, I didn't have major pain. I mean, it was swollen. I tried to treat it so to not have any kind of like thrombosis or whatever. I did all of that at home. I mean, even going to the hospital was like it wasn't worse the bother somehow, and I could fully move it because I had an extra joint. So I thought everything is fine, but and I could even lift things with it. So basically, the injury didn't feel as critical as it was. And then when the doctors told me, okay, I can maybe start doing some sports again, I fell off a skateboard, and then the thing just really went like it pulled itself through the joint, and then it really needed fixing. And then the surgeon was actually quite shocked, and he's normally not easily shocked because he's working with motorcyclists.
Sorry,
it was not my best hour.
Just that must have been really frustrating though, because as you say, keeping fit is very important to you.
Yeah.
And keeping fit ended up actually meaning not being able to keep fit for a significant amount of time.
The best part of it that I actually, of course, also like telling, and I'm sure I told it to you actually while running. I remember in Turku. Is that of course it was very weird that I didn't have pain or that I was fully functional for 2 months before actually really having to treat the fracture, which kind of like also gave myself I gave myself some kind of tough guy image. And then after the cast came off, after they had fixed the elbow, and I couldn't even touch my chin, I couldn't shave, I couldn't reach. So I went to physio, and I told the guy, because I was at this point I was really scared because I've had no threat by this injury. So I just thought, okay, that's really bad if your body doesn't tell you that something's wrong. So I told the guy that he has to watch out because I don't feel any pain. And then I cried like a child because it was extremely painful. I feel all the pain that one needs to feel. I was just for some reason lucky that the fracture itself wasn't painful, but it was the rehab was really, really hard.
So I shouldn't laugh. Just I thought don't worry.
Laugh about it.
It's not going to hurt. You can't hurt it. And then in tears moments, physios, don't you hate them sometimes? Oh. Okay, it's some quick fire questions for you. Oh, Finland or Germany?
Finland. No, not Germany.
Oh gosh, you
there's
realize that you could get kicked out of Germany in that quick fire answer. Spain or Finland?
Both. If I have to choose, no, Finland has a drawback, I would say, just because the winters are very harsh. I never experienced them. Just picking the good part is not enough to I think Spain, it would be Spain. Spain is a good all-year country, and Germany.
Hey, as a budding astronaut, the moon or Mars?
Moon.
Ooh, okay.
I mean, as in science, do things by steps.
Star Trek or Star Wars?
This should be a longer answer. First of all, I would have to say both because I was a huge fan. Now, in retrospect, I would say Star Trek.
It's the right answer. There's more science.
I did my school English paper that was part of my final examination on Star Trek, and I got full grades.
So we can thank your top grades to Star Trek. What's your favorite film of all time?
This I cannot answer because it's hard for me to rank to rank quickly. It's not quite the right answer, but I would say anything from James Cameron I find normally deeply fascinating because of the level of craftsmanship that goes into it and the way that he can reinvent stories. So meaning whenever something new comes out, I really appreciate the craftsmanship. Okay. That's maybe the closest that I can come.
So what's your favorite Christmas film? Because he hasn't done a Christmas film.
It was Christmas film. Maybe in Germany, when I was a kid, they showed classics. Could have been. I think I saw Ben-Hur actually around that time. This was used to be a Christmas film in Germany. Maybe it's an association. I think they're better Christmas movies. I actually saw you ask this question to Jean-Yves, and Die Hard is not my answer.
I like Die Hard as well, though.
I do, but actually that's okay. That's a Christmas movie. Let's agree, yeah.
Are you an early bird or night owl?
I used to be a night owl. Absolutely, like during the marine biology courses, during my studies. Basically, while working with quota, I mean night owl. But I'm transforming to an earlier bird because it's giving you more out of it's giving you more of the day.
PC or Mac?
PC.
McDonald's or Burger King?
McDonald's, because I was growing up next to one of the first, I think maybe the first McDonald's in Germany, and so it's a childhood memory.
Okay. Coffee or tea?
Coffee.
And what are you drinking? You got a coffee before this, so what sort of coffee did you get?
These days I drink like an espresso with milk. So I drink it in any form. That's the thing you learn when you work. I mean, I used to work in the hospital and doing night shifts, so I normally just drink filter coffee straight and without sugar, because then all you need is have to find the filters in the coffee. You don't need anything else, no milk, no sugar. When I moved to Spain, I actually started drinking cortados, so cut milk shots. And ever since then, I tend, when I find it, to put milk in it.
Okay. Beer or wine?
Wine.
Oh, see, that wouldn't have been the answer 10, 15 years ago. So red or white wine?
Red.
God, what has Spain done to you? No, it's a good answer.
Yeah, well, it's not the Spanish wine. I actually was really suffering from it. So I started to appreciate wine while I was still in Germany, partially with quota, partially with people we met at like at EMBL. And I really got into Italian wine. And then you go to Spain, where, of course, countries are quite protective of their own produce. So the only thing that you could find every now and then was Lambrusco. So meaning that didn't work at all. It got better. And now in Germany, of course, since they don't have that kind of stake in the red wines, you find any kind of wine. And I appreciate the complexity of the wines.
Okay. Chocolate or cheese?
Chocolate.
Milk or dark?
Dark, actually. That's my brain speaking. Actually, I think the inner child says milk chocolate.
Yeah, my work bag, I'm just reaching down to my work bag, is always has different chocolates in it. I'll stop there before I embarrass myself too much. It's getting ready to go off to Bina and Leiby. So I've loaded my chocolate and just hope it doesn't melt when I fly over, or when I land. Actually, it's less than when you get back to it. What is your favorite food? If you could choose anything, so if you were to be taken out, what is your favorite food to choose?
This again, I have trouble with ranking. If I think about the things I cannot easily get, I would say maybe some Japanese food in Japan, because there are some things you would just not be able to experience otherwise. And I think I had some very, very nice different dishes there. Yeah, maybe that.
And is there any food that you dislike?
I have a very strong. The thing is, I'm not allergic against anything, so I can eat everything. And in family context, to be polite, to be a good guest, I eat everything, and really everything. But I don't like cucumbers nor tomatoes. So the classical summer vegetables that people find refreshing, somehow it's probably even physiological. There's some connection between those, and it's saying something, I think, about the way that that your digestion works. If you don't like them, I don't. I can eat them. I don't break out in rashes or anything. I just don't like them.
Okay. And who cooks at home?
Mainly my wife, and she's doing a great job because she's also diversifying. She's cooking different styles. I do some dishes well, including some pastas. I mean, I do some basic dishes as well, and now that people like me doing them, and then I can be useful.
Okay.
Or meat. I can do a good roast or some pasta variations. I'm good with those. Other things I leave to the experts.
What's your favorite color?
Actually, blue.
Looking at you in a slightly blue top, and you've got a blue filing cabinet behind you. Yeah, that didn't surprise me too much. By the way, did you actually deliver? Oh, you've got a whiteboard. People always got Timo sitting there in his office with a whiteboard with lots of equations on it. Did you put those on it just so it looked really smart for the interview?
I had Sheldon put them on. No, I really enjoy the whiteboard. Well, actually, I should press it in front of that because I think there's an error there, and some of the questions are really very naive questions that I wrote down many years ago that maybe a physicist would be not really impressed with. But no, it shows my interest in physical chemistry. That is, I think, now my driving. If I think about the things that I'm interested in on the development side, I would say. I have for a long time been interested in physical chemistry, and I'm starting to approach the language that is needed to understand it more and more. And that is not where I started out with, because it was always like studying biology. It's definitely not a focus. And after immersing myself in optics, I realized I gravitate over the last years towards fluorescence and physical chemistry.
Okay. Last quick fire question, I think. Favorite conference?
ELMI.
Again.
Or simply for the, I mean, I think we've been on board for a long, long time. Actually, I've been there from the beginning on, and I like that it takes the companies on board so much, because we all, I mean, as persons, we are all coming from the same place and we share a deep interest. And I think this is the place where it was the best realized, that you just really do this. It's not just one side and the other. Yeah, ELMI in all its forms.
And thinking of bringing the companies along, your current role, in 30 seconds, what is the sort of strapline? What is your current role? Because it's a bit different to other facilities.
Yeah, okay, 30 seconds is hard, but no, it's actually true. So I worked in facility for a long time. I mean, I was in Spain, in Barcelona, for whatever, in the end, almost 15 years. So I think I understand how to run a core facility. And a core facility has the main purpose must be to work well inside the own institute. Anything outside is a bonus. The imaging center where I'm working now at EMBL is not the ALMF. It's not the Advanced Light Microscopy facility. I mean, the ALMF will have its 25th anniversary this month. So next month, this will be a major celebration. I will be very happy to be part of it. And it's still needed as in the first day. I mean, core facility is absolutely essential. The imaging center reflects this additional role of EMBL to be open for external visits. And this is a different business model, as you know. I mean, you also have external visitors, but the logistical effort is not something you do on the side, and this is very much dedicated. And also, what we are aiming for is to connect earlier on to the technologies by also going into the pre-commercial field, which is something as a facility that I never could manage. I mean, in Spain, I just didn't have the possibility to support the access to such a system. Here, we're basically building them up. So it's a very different role, and I saw it as a interesting progression out of something that I think I understand, and I also appreciate why it has to be different. That's why I was actually interested to come here.
And your team's quite big over there.
Actually, well, no, it's two teams.
Yeah, joint team.
So, and it's a bit dated. So there's now more people. It has grown. So these are two people. So actually, most of the people that you see there are actually from the cryo-EM team of my colleague Simone. He's the bearded Italian in front with a blue pullover. Exactly. So he's basically my counterpart on the cryo-EM side. But the imaging center has two sides: light microscopy and cryo-EM. And of course, what we want to meet in, and where we're basically now meeting, is in correlative approaches. So many of the people are, for example, also cryo-EM specialists, but there's also my application specialists on the picture. I cannot point to them, of course, right now. There's one of the two optical engineers. The other one had not started at this point. There's a data analyst. So I have a team of 5 for the service side. So 2 application specialists, 2 optical engineers, and a data analyst. And it's similar on the EM side. And there's also research on both sides. There are also some research positions from Timo in this case. Mine are not on the picture. They started this year because I now also really want to go into massive development. But it's a great team.
Beyond this, you have quite a lot of interactions with commercial companies as well, which means your job is, it's not just being a scientist, is it anymore? The role is more than just being a scientist. You have to be a manager. You have to be a sort of semi-businessman as well. Would that be fair to say?
To be a businessman of what? So
to have a business aspect to you, looking at the finance, you've got the company engagements, the legalities around that, plus the science. Quite a lot of skills that you're holding there.
Well, I think actually, and I mean, I guess you wrote the book on that one yourself, or you could write a book on this one if you haven't written it already. You have to go beyond the technology itself. If in this, or there's a lot of opportunities to interact in forms that are very, very beneficial. But for this, you have to also understand exactly these additional challenges. But it's not only about where's the on button on the microscope or what do you have to optimize experimentally. It's about how to embed technologies, how to play them to their strongest point, how to actually interact with companies also in the form of a mutual beneficial relationship. So not trying to maybe just like sell, okay, place it here and just looking maybe for a bargain, but thinking about what does it actually take, and not only with companies. It's the same for any kind of, let's say, networking activity. Where are the points where you both walk out as with a benefit, and try to work for those? But this goes beyond science. This is now becoming, of course, partially management things, partially politics. I mean, that's something I did for many years, also in the context of trying to network for Eurobio imaging. And these things you pick up. You ideally also take courses in. Courses are now more and more available. And I think we do need, but this is the point. I mean, when we finished studies, this was just the beginning. So anything picked up afterwards, either you do it yourself, or ideally you actually cut the corners and you get target trainings in the things that you need. Our tool set is much bigger than just the technology. And this is when you work with companies, when you work with balancing different aspects in major undertakings. That's if you want to work with multiple partners and respect all the interests, this is what you need.
I think when we started out, those courses didn't exist, but our roles didn't exist particularly.
That's the point. I mean, 25 years of ALMF, they were some of the first, and this is basically when we started.
So we are nearly up to the hour, and I have 2 questions I still want to ask. Can I ask you, what has there been any time in your career that you found particularly challenging or difficult for whatever reason? Everything at the moment sounds serene, but there must have been bumps, challenges, uncomfortable times.
I'm sure there were. Yes, I remember sometimes, but I would say these were isolated incidents. Some in retrospect funny. I mean, like this one night of shock that I had when I was writing up my PhD thesis, and when I was writing the methods, I thought I had used the wrong plasmid. I mean, like in the middle of the night, at the time when after you already published 2 papers on this thing, you kind of like just try to cross the T's, and you find something that you consider a fundamental error. That was quite shocking. But this is like this thing that throws you for a loop. But it was done within half a day. I came in the next morning, we looked at it, and we realized that the nomenclature changed. So meaning these things very often dissolve themselves. You can still feel this initial shock. Later on, I think with some incidents, maybe that's also where one can learn thoroughness. I once had an incident with a system where I really thought, okay, I mean, it just broke, or the ceiling broke in front of my eyes, more or less, and damaged the system in my eyes severely. And I thought that's the end of me. And I can only deeply appreciate that the persons I was working with on the institute side were actually very thorough, had done all the right insurances and everything. So everything was in the end fine. But this moment when I realized that I possibly had not considered all the options, and I saw my career ending at this point, and that took maybe 2 or 3 days to walk off. It actually also brought me in a very nice direction, because I was so upset with the incident, and that first of all, I think I learned for the future to be much more respecting the process. So it's not only about getting what you can get, but also getting it in a way that everybody's protected. But also, I just realized I have to move my facility into a new location. I was really fed up, and that gave me the drive to then develop. It took still more time, but to develop, let's say, a relocation of the facility, which I at this point really wanted. So meaning something good can come out of it. But of course, I felt like, yeah, I was not in a good place then. These things I remember, very few of those.
That last one's a good point. You know, when you look at the cost, certainly in the UK, we had a postdoc has a cost associated. It's not just their salary; it's the overhead, which is the estates and the infrastructure around them. And what you just said, it was the infrastructure that's being put around. That someone was there, they sorted the insurance, they're there to help with it. You know, the people that, for a lot of scientists, they're the hidden people that we don't see because they're not in our departments. But my goodness, we need that infrastructure behind us to enable us to do our work with that layer of protection as well.
Yes, and one can also see this maybe as a restriction. I mean, of course, I think maybe also you are also, of course, sometimes bad-mouthing some things. And I mean, and not everything that's coming out of a structure is actually helpful. I mean, we also are faced with the flip side. But no, I mean, the point is we are maybe in public research, at least one sometimes forgets the kind of amounts of money that, I mean, you're making calculations for what a running cost is. I make calculations for what a running cost is. These things are massive. This is not what a person sees that comes on the confocal microscope. But behind all of that are incredible amount of cost of a postdoc, cost of any position. These are massive amounts of money that need to be properly handled. And sometimes it will not be in your interest. But I mean, the thing is, imagine a world without this management and you will basically have no research.
Yeah.
I still want them to get better, but yeah.
I've said it before, but at York, touch wood, it's never got in the way. Usually the system is there to help, and where there's not a system in place, they tend to help us put it in place to help us move forward. Yeah.
I mean, I can agree basically with this. Most of my experiences have been good. I mean, I remember maybe the most frustrating aspects I've experienced was still at university, where maybe really grown structures sometimes are quite noticeable. But I've otherwise always been in places with very flat hierarchies, and I have never encountered, maybe I'm lucky in this, never encountered anybody that just didn't want to go along for whatever reason. I mean, they were all reasonable people. And there are reasonable people I'm dealing with.
Yeah, I think the same this side. And the final question: what's been the most fun? When, if you could relive a year of your career, when was the most fun time to relive?
The most fun time, probably, like, I mean, I can say I deeply enjoyed my PhD and also time at EMBL. But I mean, this is, if I hope everybody does, honestly, because that's the time when, of course, you experience many new things. I learned many things late in life. Like, I only started scuba diving when I was in my 30s. I learned snowboarding basically when I started my PhD together with Kota. We started on the same day with different results. He broke his hand. That's actually why on the picture, the next year, he's actually trying out hand supports, because one learns from one's mistakes. And then also the time at EMBL was really, really nice. Everything afterwards, everything before was also great, but in different ways. I mean, you're only young ones. And I think I enjoyed that. And
I said that as one last thing, but you sent me one more picture, and I'm intrigued because I have no idea what's going on here. This is a picture. Please describe the picture for those listening, because I don't know what we have here.
Yeah, that's also why I included it. Actually, maybe not the most. So this is actually just this was a test shot taken with a Polaroid for an interview for a German magazine. where they asked somebody from the studentship in Munich, and I was the representative, to explain what a biologist would like to be, so to explain the job of a biologist. I mean, this is of course ridiculous. I mean, there's so many different things. And for some reason, they wanted to connect this to paleontology. And this was also around the time of Jurassic Park. So all of a sudden, dinosaurs, so you see some dinosaur skulls in the back, and you actually see some mammoths or mastodon like tusks even in the back. So this is the collecting or the seller of the paleontology collection, where we had an interview about one possible job field for biologists. The way that it came out was like me saying that I want to be a dinosaur researcher, which is a bit naive, but yeah, maybe the other thing, if not an astronaut, what do you want to be? You want to work with dinosaurs. That was a bit of I was representing more the students per se in this. But it was a very strange.
And I had to show this one because this is Timo sitting in front of a microscope, very famous in Japan. All Japanese. I have no idea what the Japanese writing says. I didn't get Google Translate on it, but see, fame everywhere, from the paleontology dinosaur days through to being a top scientist.
Seems like I'm always putting myself in the picture. If one selects it. Now, this was a funny. When I was visiting Japan, I was also visiting like a headquarters, and since I just worked a lot on this is a SP2 AOBS system for spectral mixing. Actually, this was something I was working on then. They took that photo in a very professional setting, but actually the lab coat was way too small. I mean, a Japanese lab coat, so I had to really angle my arm so that it looks convincing. And it was a professional shoot, but yeah, it's funny when I found that magazine again. So yeah.
And it's P2 that you taught me on to start with.
Exactly. It's it's a very similar system to the one we were working on.
Yeah.
It all comes back.
All back round full circle. On that note, we are just over the hour. I have to say, Timo, thank you for my career. Thank you for guest starring on the Microscopists as well. Please, those who are watching, listening, please subscribe to whichever channel it is. There's lots on the back catalogue for you to go watch and listen to. But this one's been a personal indulgence to have Timo as a very special guest. It was very special to me. Timo, thank you.
Thanks so much, Pete. This was a real pleasure and honor. I mean, I've been following your career, and I'm deeply impressed. And therefore, it was really an absolute joy right now to actually, take stock a little bit for both of us, and and continue doing this in the future.
And see you soon. Thank you, everyone.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.